All right, this is our last lecture on chapter 3, which is one of the really most important chapters because all living organisms are made of cells, and so this is our introduction to cells. And here, what I plan to do is go through some examples and some applications, and that kind of talk about the importance of this and what this led to. So, first, I'm going to go through a couple prokaryotic organisms. These are simple organisms like bacteria and archaea. Those are the two domains. And we really kind of recognize these as different pretty early because they were much smaller than the cells we'd find in plants and animals. But it wasn't until we really had a good look at them and had seen a lot of them that we recognized their importance. And probably their first importance was that it was recognized that some of these cause disease. And so we had long known that people that were ill had microorganisms in unusual places and sometimes large numbers of them. But it really took finding them in wound sites in specific regions and then experimentally infecting people and animals. And so in the late 1800s, when microscopes were widespread and people were starting to develop the techniques, it was recognized some of the most important human and animal diseases were caused by bacteria. Probably one of the greatest ones was tuberculosis. Tuberculosis was known to cause respiratory and other diseases in areas where people were crowded and had poor sanitation and poor nutrition. Eventually, they recognized that tuberculosis could be transferred with the transfer of bacteria. And that recognition happened by a famous biologist, Robert Koch, and they still had no treatment for it, but he recognized that if we isolate these people, that tuberculosis would stop spreading. And if we took care of them in like a sanitarium where we gave them good nutrition and allowed them to rest, that often their immune system could recover it. Until about 1940, we really didn't have any treatment for bacterial infections. We discovered that by accident. This is Sir Alexander Fleming there in his lab. He was growing a disease-causing bacteria called Staphylococcus aureus when he noticed that one of his plates, which had been accidentally contaminated with a fungus called penicillium, had an area where the bacteria couldn't grow. That the penicillin mold colony was secreting something that inhibited the growth of bacteria. This was eventually purified and concentrated, and it was recognized that it was very toxic for bacteria, but not for people. And so this was our first treatment for bacterial infections, penicillin. Prior to that, all bacterial infections were untreatable. And so this is a molecule that attacks a feature of bacterial cells that our cells don't have. We don't have a cell wall, and we don't make the materials that those bacterial cells make. So this drug that interferes with that is non-toxic. Okay, now let me switch over and do some talking about some eukaryotic cells, our cells, red blood cells. These are kind of unusual cells because in our blood they have a very specific shape, kind of a flattened disc with a hollow part in the center. And they're unusual in that as adult cells, they don't have a nucleus, and they're almost entirely filled with one protein, and that protein is called hemoglobin. These cells are made in red bone marrow, and they are made in response to a hormone. The cells eventually die and are recycled, but before that, they play a big role in the blood, transporting oxygen and carbon dioxide. Here's another picture. There are some white blood cells there, but by far the most abundant cells in our blood are these red blood cells. Okay, so what's unusual about these cells is they lack a nucleus when they're made, or when they're mature, but when they're made in the red bone marrow, they're made from stem cells that have a nucleus, and these cells produce an unusual protein called hemoglobin. It's actually four protein chains that are linked together and hold some unusual structures called heme units. And as these cells mature, which you can see down in the corner here, the lower right corner, they gradually lose their nucleus and they accumulate one protein, hemoglobin. And this hemoglobin is really crucial because it has these heme units with iron and nitrogen in them, and these reversibly bind to oxygen. And each hemoglobin molecule can carry four oxygen molecules, and there are literally millions and millions of these in each red blood cell. This is what allows our blood to rapidly pick up oxygen in the lungs and then carry it to our tissues. Red blood cells can't be, they can't reproduce as cells. They actually have to be made from stem cells. These cells are going to be recycled in the liver and spleen. As they get old, they'll be broken down and the hemoglobin will be at least partially recycled. Some will be excreted in bile, but the rest will be sent back to be made into new red blood cells. Okay, so how do red blood cells affect us? Well, it's really important to know that blood is one of the most crucial tissues, and blood supplies our tissues, our other tissues, with oxygen and gets rid of carbon dioxide. Even a relatively minor loss of blood can be really, really serious. It causes what's called circulatory system shock, or just shock. And that's not an emotional shock. What it means is that if there's blood loss, blood pressure falls, and the pulse becomes weak and rapid because the heart is trying to get the blood as quickly as possible to somewhere else. But a lack of oxygen can cause organ failure, okay? And that's because this is anoxia. And if this goes on for any length of time, this is potentially fatal. And so it was well known that blood loss was a really serious thing, and there really wasn't much you could do about it until we developed blood transfusion. Accident victims, wounded soldiers, even complex surgeries were all likely to be fatal because of the loss of 2 liters of blood was too much, and that would kill you. It wasn't until we found that we could transfuse blood. Actually, it's a transplant from one person to another. But this was complicated by the appearance of blood groups. Okay, all blood contains antibodies, molecules that react to foreign molecules that bind to them and destroy them. These can inactivate bacteria and viruses, but also if you get a transplant or a transfusion that doesn't match your tissues, you're likely to have antibodies against it. And what these antibodies do is they recognize surface proteins that shouldn't be there, and then they bind to them and they'll destroy these cells. And so there are these blood groups that type blood by the type of molecules on their surface. And so there are 4 major blood groups, the O, A, B, and AB. And then in each group, you also have Rh, positive and negative. And with these blood groups, if you mix the incompatible blood types, what happens is the red blood cells will be clumped together by antibodies binding them. And if this happens in your blood vessel, it will kill you. And so it wasn't until we could type blood that made blood transfusions possible. And these blood groups are really crucial to understand. And about approximately half the people are about one blood group. They are the O group. But here you see the A, the B, ABs, which are relatively rare, and then the O, which are very common. We used to use this as evidence in murder trials because you could rule out some people if their blood type didn't match blood found on the scene. But even if it matched, you couldn't say for sure that was them because it could be somebody else that had their blood group. We really couldn't reliably identify individuals until we could start to identify their DNA. Using DNA, we can find unique sequences in each individual or unique patterns of DNA that can help us identify people much more specifically. Okay, the last example I'm going to talk about are viruses and how viruses infect cells. And this is true for influenza virus or COVID virus or the cold virus, any virus. But the example I'm going to use are helper T cells and the HIV virus. HIV is human immunodeficiency virus, and it infects basically one or two types of cells in the human body that have the right type of molecules on their surface. HIV infects these cells and kills them. These are called helper T cells or helper T lymphocytes. And as HIV starts to decrease the number of these cells in the blood, people develop more and more symptoms of immunodeficiency. So if normally we have about 1,000 of these cells in each milliliter of blood, if HIV reduces that to less than 200 cells per milliliter of blood, we have AIDS. We will have the symptoms of immunodeficiency. So how does this virus do this? Well, the virus has molecules on its surface that recognize some of the molecules on the host cell. And so it can only infect cells that have the right molecules that bind to the virus proteins. And once that happens, that triggers a series of reactions that allows HIV to enter cells. Okay, these molecules are usually proteins, but sometimes they're sugar. They have some sugar as well on them. And so it's this interaction of molecules that leads to the specificity of infecting specific cells in specific species. And so HIV can't transfer to your dog because your dog will have different cells on the surface of its lymphocytes. Okay, so these are just some examples that you can use when you're thinking about cells and how cells work and how cells interact and the chemicals that are found in cells, the proteins, the DNA, various molecules. Okay,